Okay, hi, good morning. Welcome everyone to the Brain Body Interaction Seminar. My name is Jerica Barron and I'm a graduate student at UCSF. And today I'm excited to welcome Tony Wyss-Coray to our final seminar of the season. So everyone, you should check out our website, sign up for our mailing list, and follow us on Twitter to stay up to date for when we return in the fall. You can also find previously recorded talks on our YouTube channel, so I'll share those links momentarily in the chat. So without further ado, I'd like to today introduce you to today's speaker, Dr. Wyss-Coray. So Dr. Wyss-Coray is the D.H. Chen Distinguished Professor of Neurology and Neurological Sciences at Stanford University. He was also recently appointed to the director of the Phil and Penny Knight Initiative for Brain Resilience, which is a new interdisciplinary endeavor to understand neurodegenerative processes. He's garnered many awards over the years, from the NIH Director's Transformative Research Award to the Pioneer Award, to even Time Magazine's 50 Most Influential People Transforming Healthcare. His lab studies brain aging and neurodegeneration with a focus on age-related cognitive decline and Alzheimer's disease. Among many achievements, he's led discoveries of how blood-derived factors can impact brain function, described roles in innate and adaptive immunity in the aging and diseased brain, and helped lead the Tabula Mura single-cell atlas project. So Dr. Wyss-Coray has also worked to translate some of these incredible research findings into therapeutics. He's also co-founder of AlkaHest, a company developing plasma-based therapies. So I will stop there and hand this off to Dr. Wyss-Coray. Thank you so much for being here today, and we look forward to your talk.
Thank you, Jerica. Thank you for having me. I'm excited to be here and tell you a bit about what we're doing and what motivates us to try to understand brain aging and neurodegeneration. Here, a couple disclosures. So as Jerica said, we're interested in understanding how the brain changes with age and why it becomes susceptible to cognitive dysfunction, neurodegeneration, and one of the most devastating diseases, probably Alzheimer's disease, where people lose sort of their own self. And Alzheimer's disease is not the only age-related disease, as you know. As we get older, as this graph shows here, people have more and more age-related diseases. And it's estimated that at age 65, people have at least 2 or more conditions that are clearly driven by age. And these could be heart disease, diabetes, and so forth. Most of them have a very strong aging component, meaning that you don't get them when you're young. That has prompted a lot of people, or an increasing number of researchers, to argue, why don't we try to understand the aging process to intervene in these age-related diseases, rather than treating each one individually and finding disease-specific processes? What if we could find common processes related to aging, slow down the aging process, or even reverse it in what people often refer to as rejuvenation? And this is really not a pipe dream anymore. At least in animals, in mice, there's now multiple interventions that can slow down or even reverse aging. And these include metabolic interventions such as caloric restriction, exercise, the use of targeted small molecules that intervene in metabolism, the removal of senescent cells, or a really hot field right now is the transient reprogramming of cells, where you transiently activate the Yamanaka factor or similar transcription factor that if you turn them on constantly, they would transform a cell into an induced pluripotent stem cells. But if you turn them on only for a few days or even a few hours, what happens is that the age-related epigenetic marks on the DNA get erased. And the cell keeps its identity, but it essentially becomes younger. And has been shown now by Juan Carlos Belmonte, for example, and others, that this leads to a functional rejuvenation of tissues, and entire organism in mice. And then treatment with young blood or even spinal fluid, as I'll discuss in my talk a bit later on. So one recent study that we carried out, and this was actually done by Tal Iram, a senior postdoc or now an instructor in the lab, who argued that since the cerebrospinal fluid, so this is the fluid that bases the brain, since the composition of that fluid changes with age, what if we could reverse or change the composition of that fluid and basically infuse young CSF into an old brain cavity, into an old mouse? Would that have a rejuvenating effect on the brain? Sort of changing the systemic environment of the brain directly, would that have a beneficial effect? And so what Tal did, she collected CSF from lots and lots of mice and then infused it slowly via an osmotic mini pump into old mice. And what she found is that if she did this for about a week, this slow infusion of young CSF, she could improve memory function, specifically in a fear conditioning memory test, the recall to a previously exposed adverse environment. She could recall that, the mice could recall that better if they were infused with young spinal fluid compared to an artificial control. And when she then looked at how does this infusion affect the gene expression across the hippocampus, she found that the cells that are most responsive were of the oligodendrocyte lineage. So those had the most gene expression changes following this regimen of about 1 week of infusion and then taking them down right after. And what you see here is an illustration of some of these signature genes that are transcription factor of the oligodendrocyte lineage, or specific genes that are characteristic of these cells. She could then, in close collaboration with Brad Zucchero here, show that there is an increase in proliferation of the oligodendrocyte precursor cells. So there's more of these cells being generated in vivo. And when we culture oligodendrocyte precursor and expose them to young mouse or young human CSF, that led to an increase in differentiation and maturation of these cells, suggesting that there's factor in the young CSF that both in vivo and in vitro can induce this oligodendrocyte lineage to generate more cells and make more differentiated cells, something that the young brain can do in response to a memory challenge or a learning paradigm, but in old mice, that capacity has decreased. And the function of these cells is a very specialized one for those who don't work in the brain. These are the cells that basically wrap the axons, the connections between nerve cells. So they produce the insulation that allows efficient and fast transmission of an electrical signal from one cell to another. And again, with age, the capacity of these cells to respond to a change and to be so-called plastic decreases, and with the young CSF that could be restored. Tal then followed up to try to understand what is the molecular basis of these effects. And in a nice commentary by Maria Latinen and Savatsky, they show here that the CSF induces these oligodendrocytes to activate a transcription factor that is called SRF, in part through the secretion of a growth factor called FGF17. And so there's a whole cascade that leads to activation of these cells and restoration of function. But sort of in a broader context, as I mentioned earlier, the question of are there factors or can factors in the environment influence the function of cells or the function of stem cells even um, was addressed a few years ago now by a good colleague of mine who just recently moved to UCLA, Tom Rando. And Tom was specifically interested in stem cell aging in the muscle. And he asked a specific question: do these stem cells get exhausted or don't respond to a challenge with aging because they lack an intrinsic signal, or is it because there's something in the environment missing? And so Tom used this old model called parabiosis, where you essentially suture two mice together at their flank. That leads to growth of vasculature of blood vessels into the wound, and they eventually fuse and allow now for an exchange of blood from the young mouse to the old and vice versa. So you have a slow diffusion of blood, an exchange of blood between these two mice. And that allowed Tom to ask the question again: do stem cells in the old muscle, do they respond to circulatory cues from the young mouse? And can they potentially be reactivated and rejuvenated? And indeed, that's what he found. These old stem cells reacted almost like young ones to a challenge, to an injury, for example. He also reported rejuvenating effects on the liver, and he noticed that there's an increase in proliferation of cells in the brain. And that was the basis of a collaboration between his lab and my lab to really understand the effects of young blood, of a young circulatory environment on the brain. And in the meantime, many other labs have studied the brain and many other organs and showed that indeed young blood can rejuvenate these tissues and organs. What we were also able to do is to show that you actually don't need parabiosis. You can simply collect, as we showed with the CSF paradigm, you can collect blood or just a liquid fraction called plasma from young mice and repeatedly infuse it into old mice and reproduce many of these effects. And this was really pioneered in my lab by Saul Vileda when he was a graduate student. He's now an associate professor at UCSF. And what Saul was able to show is that if he infused young plasma repeatedly into old mice, that led to an increase in stem cell activity in the brain, increased synaptic plasticity, as well as improvement of memory function. And then others in the lab, including Hannadi Youssef, showed that it also reduces brain inflammation, particularly in the vasculature. The opposite was also true, as Saul was able to show, is if you take plasma from old mice and you infuse it into young mice, you get the opposite effect. You reduce stem cell activity, you reduce synaptic plasticity, memory function, and again, studies by Hannadi Youssef and Michelle Chan, together with Steve Quake, showed that there's more inflammation, and particularly more inflammation in the vasculature. A number of labs have tried to identify what are the key factors. I list some of the positive ones here in green, as well as negative factors that mediate sort of the detrimental effects of age plasma in red, such as CCL11 that we followed up a bit, as well as beta-2 microglobulin, and we're still very interested in VCAM1. Now, because this is so translational, as Cherika mentioned earlier, we started a company called Alcahes and worked particularly with a company called Grifols out of Spain. They're one of these plasma companies that collect plasma from thousands of donors every day. They pool this and make different fractions. So if you go to the hospital and you have surgery and you lost blood, then usually you get only a fraction of plasma. You get often an albumin-rich fraction. If people with immunodeficiency, for example, after a bone marrow transplant or autoimmune diseases, they often get an immunoglobulin enriched fraction, so IVIG. So they make a lot of these products. And together with them, we tested different plasma fractions, and we were able to show in a small clinical study here at the bottom that a plasma fraction has significant benefits when we compare it to historic Alzheimer's disease placebo control. So this was only a small trial. It was not powered, but showed promising results. And then Grifols actually carried out a much larger study in 500 patients that was placebo controlled, double-blinded, so-called phase 2b, phase 3 trial. And here Alzheimer's patients were first, they removed the aged plasma in what is called apheresis, and then infused them with a albumin-rich fraction. And because their average donors are around 30 years of age, that is essentially a young fraction of plasma. Now, albumin still contains hundreds of other proteins. It is a carrier of many growth factors. But what's really exciting is that this trial was actually positive. It showed a benefit on disease progression, memory, as well as daily functioning in moderate Alzheimer's disease, suggesting that the findings from our mouse studies translate, at least in these early studies, to humans and seem to have beneficial effects. Now, this all seems almost too good to be true, and we really want to understand how does this all work. And there's, of course, a lot of questions that you can ask. So how does young blood potentially rejuvenate the body? How does it rejuvenate the brain? What are the factors? I showed you some of them, but we don't think that it's just one factor, certainly not. But we also think there's many other factors that have not been discovered yet. Where are these factors produced? Why do they change with age? Which cells are actually responding to the factors? Are all of the cells responding, or and which ones in which different cell tissues? With respect to the brain, how would these factors in the blood even get to the brain? There is this blood-brain barrier that people always invoke that we were taught that doesn't let stuff into the brain. We know now it's much more sophisticated. There's actually lots of plasma proteins that get into the brain. I don't have time to talk about this, but still that's a big challenge. And then sort of more broader question that I just hinted at, if cells and tissues respond differently to young or aged plasma, do actually all cells and tissue age in synchrony in an organism, or how do they age? And then directly related to that is, how do we even measure the age of a cell or of a tissue, especially in a living organism, in a living person? So Jerica mentioned this briefly, but one question that we had, and we approached sort of quite naively at the time, was where do the proteins that we had identified, where are they being produced in the body? And this is a study that Nick Schaum, when he was a graduate student, tried to tackle. And our naive approach to this initially was, let's just collect all the major tissues from young and old mice and look where are the growth factors, the cytokines, the chemokines produced, in which tissues were they produced, and how do they change with age. And we started to talk to Steve Quake, who just became the co-director of the Jen Zuckerberg BioHub. And so he was very excited about this project, but he also had a lot of resources and decided that he was interested in doing this together with us. And so we recruited more than a dozen different faculty with expertise on all these different tissues in the mouse, and started to take down mice at different ages, mice that underwent parabiosis, isolated their tissues and associated them for single cell RNA sequencing. And that led to these different studies that gives you an idea of which gene is expressed in which cell type across the organism, and then how do they change with age. And what we saw throughout the study when we looked at aging is, I think a key take-home message is And it's not surprising, probably. Aging is definitely not a linear process. And this is illustrated here, for example, in gonadal adipose tissue, where we sort of show graphically the changes in gene expression from blue low levels to red high levels of a gene across the age from 1 to 27 months of age in mice. And you can see these dramatic changes that occur in a non-linear fashion, but really also throughout lifespan. And you can really argue, when does aging actually start? And you could certainly defend the position that it starts whenever a cell is born, that cell starts to age. And so you basically start aging while you're born. And this can be illustrated here across all these different organs, downregulated, upregulated genes. And again, you see that different tissues show individual aging trajectories. You can see the same thing if you look at the cell type level. And again, not synchronized. And we're not the only one to make that conclusion. This is probably the most comprehensive study, but people have noticed that over the years, looking at individual cell types in all kinds of different animal model systems, that aging is asynchronous at the tissue and cellular level. And so you can sort of show this more broadly when you look, for example, in an organism from development to the reproductive phase to post-reproductive phase. While people in the past have often sort of studied aging only in the post-reproductive phase, we now know that age-related changes really occur throughout lifespan. There's different tissue that show their most dramatic transcriptional changes at different times throughout lifespan, as illustrated here. So when you then look at these gene expression changes, you can actually assign some genes to specific cells and tissues, and I come back that at the end of my talk. But you can also start asking now, which cells do, for example, respond to a change in the systemic environment, such as through parabiosis. And so here we use this model parabiosis, and to just orient you, we typically have a pair of young mice. These are called isochronic. We have old mice, old isochronic. And then we have the heterochronic, so a young with an old. And that allows you to really make multiple different comparisons. But for simplicity, what we typically do is we look at normal aging, sort of as our ground truth, shown here as age in red. And then we compare how does a young mouse exposed to an old environment? So how does that accelerate aging, if you will? So we call this accelerated aging. A young environment, a young organism exposed to old. Does that mimic normal aging? So we call this accelerated aging. And then you can ask the opposite question. How does the old mouse experience the exposure to a young environment? So does that reverse aging process? So if a gene went up with normal aging, does it now go back? We call this rejuvenation. And you can imagine, so if you have normal aging here, that accelerated aging mimics some of these effects, and rejuvenation reverses some of these effects. And that's what we look at here. So first, normal aging. This is just one way to look at the data. There's many different ways, but we just count the number of genes in each one of these cell types. So we had about 50 different cell types in this study, that we had enough statistical power. So just looking at normal aging, you see, for example, hepatocytes, stromal cells, macrophage, lineage cells show the highest number of genes that are changed with aging. But really, almost every cell shows thousands of genes that change from young to old. When you look at accelerated aging, you see still in some of these top cells, you see more than 1,000 genes that are changed as a result of exposure simply to an old circulation. So factors that are derived from this old mouse that reach the young organism, they induce thousands of changes in these cell types. And then the same in the rejuvenation paradigm. So if we look at the old mouse exposed to factors from the young mouse, again, hundreds or sometimes thousands of genes that change. And you see the rank order is different. So here, for example, hematopoietic stem cells seem to be the most responsive to young blood. And then you can ask, as I mentioned earlier, which ones of these genes in the accelerated aging show the same direction as normal aging? This is shown here. So you see in these stromal cells here, you see almost 40% of the cells going the same direction. You see this also in fibroblasts. Hematopoietic stem cells seem to be quite responsive. So a significant number of genes go in the same direction, suggesting that the old factors that reach the young organism mimic really the aging process when it comes to these specific genes. And then much fewer genes, but still a significant number are reversed, and suggesting that young blood can directly antagonize the age-related changes that you normally have in an aging organism. And here, interestingly, again, hepatocytes, the top most responsive cell that is rejuvenated by young blood, and then hematopoietic stem cell and some other stem cell types here at the top in this paradigm. We can ask, what are the key pathways that are affected by this? We see that when we look at an accelerated aging paradigm in stromal cells, for example, some of the most susceptible cells to aging, the top pathway is extracellular matrix, something that increasingly comes up in aging studies, and the field has long implicated changes in extracellular matrix as a key feature of aging. Think about wrinkles in your skin. When we look at an exemplary cell type for rejuvenation, here the hepatocytes, so these are highly susceptible to young blood, and many of the genes are directly opposed by young blood, so they go in the opposite direction than what they go with normal aging. Here, very exciting, the top pathway is mitochondrial electron transport, respiration, ATP synthesis. Again, not something new, but very reassuring that a pathway that has been implicated over and over in cell culture models, in single cell organisms, in all kinds of different model organisms of aging. Mitochondria have always been implicated in the aging process. And so here we see that they're actually activated, these processes are activated in a rejuvenation paradigm that has functional benefits in mice. You can look at this more globally and ask, what are sort of common pathways of aging at the transcriptional level in an organism? And we show this here in a plot that we call a Picasso plot that Robbie Palovich, who's the first author on this study here, developed or implemented. And so here you have the different cell types that he compared, and then here you have the tissues. And what we basically show is, for each cell type, what are the top genes that change with age? And what are the common genes? So if like two cell types show the top genes are the same ones, they would have a high similarity. So a lot of genes that are changed in common. And the size of the dot shows the number of genes that change with age in each cell type. And then the lines that connect the different dots show the similarity. So if there is a line, that means their aging, the top aging signature is similar. And what we notice is sort of two features here. One is that there are cell types, such as endothelial cells, that across multiple different tissues show a common aging signature, as if these cells are aging sort of according to a more intrinsic plan of aging. The same with stromal cells. And then you have other a different aging paradigm where cells within the same tissue, such as the skin here, but of different cell types, so keratinocytes, epidermal cell, basal cells, clearly different cell types, they show a common top aging signature, as if something maybe in this tissue induces a transcriptional program. Similar here in the pancreas, where these cells seem to age in a similar fashion. And then you can also look at the same idea in the accelerated aging paradigm or in the rejuvenation paradigm. And some of these common aging signatures are preserved and some arise newly out in these paradigms, allowing to really look at new type of biology. Now, one thing you can start to do when you look now globally at an organism, how it responds to an aging intervention, to a rejuvenating intervention, is what are commonalities between different interventions. And this is a study that was just published a couple of years ago. And here they use a frequently employed rejuvenation paradigm, caloric restriction. So they had rats, young rats and old rats that received as much food as they wanted to eat. And that leads to obesity in these rats. And then calorically restricted rats. And they looked at 6 different tissues with single cell RNA sequencing. And they too found that cells respond differently to this paradigm. They found rejuvenation signatures. But what we can start doing now is see, are there common signatures, common responses between these different types of interventions? What are the key pathways that could potentially be targeted in a therapeutic intervention? So one of the big questions that I mentioned early on is, what are the factors and how do we measure aging? And they're sort of related. They're of course also related to any of these rejuvenation paradigms. While you can, in an experimental model, you can harvest the tissue, you can sacrifice the animal, and you can look whether your intervention induced a molecular readout, such as I showed you in the parabiosis paradigm, and use transcriptional changes as your sort of ground truth. You can't really do that in a living organism, especially in humans. You can only collect a certain number of tissues, such as blood tissues, or maybe cheek cells or skin fibroblasts, but that's about it. So the blood is actually a really fascinating tissue that we have been exploiting in medicine really for decades, or maybe even longer, for the fact that it really connects all the different tissues in the body. And it's somewhat trivial if you think about it, but I think it has tremendous potential to study the aging process, but to study normal physiology in general with the latest technologies and tools that have become available, as I'll discuss in a moment. So, of course, the blood carries immune cells. The immune system is very dominantly represented in blood. It carries gases, oxygen, CO2. And but it also carries information. It carries molecules that are produced by any tissue and collects these in part for catabolism, for excretion, but then it most likely also uses them to communicate between all these different tissues. And while that's sort of, again, obvious for endocrine function, hormonal function, I think this is a much broader concept that we can exploit. So one example, as I mentioned earlier, is can we study aging by looking at the composition of blood as the organism gets older? And so we've done this over the years. It's now probably 15 years, where we started with initially very simple tools where we measured a few dozen different, mostly cytokines, growth factors with array-based, antibody-based platforms, then with Luminex. And most recently, we have arrived at this platform called SomaLogic. So what they do is they synthesize tens of thousands or probably millions of different oligonucleotides with modified nucleic acids. And then they look for specific so-called aptamers, these modified oligonucleotides that specifically bind to a given protein. And here illustrated interleukin-6 binding to its cognate aptamer. This is like an antibody-antigen interaction. And interestingly, the size of the interaction is actually very similar. The footprint that where the aptamer touches on the protein is very similar to an antibody-antigen interaction. And so over the years, they developed now an array of 7,000 specific aptamers, and they will soon have 10,000. So they can measure the concentrations of currently 7,000 different proteins in essentially a drop of blood or plasma. And so in this experiment here, we collaborated with a normal aging study in Europe. It's called the interval study, as well as with near Barcelona and Sophia Millman at Einstein College. They have this centenarian study and offspring. And so we had the offspring and healthy controls from this study, as well as from the interval study, over 4,000 healthy people aged 20 to 100. And at the time, we had 3,000 proteins measured in the plasma to basically ask what is the relative concentration of these proteins in healthy people from young to old. And if you graphically represent this, you again see there are dramatic changes as people get older. So here you have the 4,000 people in the x-axis, the 3,000 proteins in the y-axis. You see low levels of proteins in yellow here. They in blue, some of them go up as you get older, and then the opposite. Higher levels when you're young, and they go down as you get old. But again, non-linear changes. If you squint your eyes, you see that even though these changes are non-linear, there seem to be sort of hotspots of changes. And what you can do is you can, at each age, you can ask how many proteins are significantly different between people, let's say, younger than 30 compared to people older than 30. And then you can add up the number of proteins that change. And what you see is that there are actually waves of aging. Yes, it's non-linear, but there still seem to be an organized change in the number of proteins that change across lifespan. Of course, this is all cross-sectional, but still, if you look at these individual, there are clearly patterns of changes. So again, what we look at here is the number of proteins that change at each given age between younger people and older people, and then we add them up. So here at age 35, you see about 440 proteins are significantly different between younger people and older people. And there's actually a lot of sex-dependent changes. At least a third or even more of the proteins are significantly different between men and women. And when I say significant, these are not tenfold differences. They're often very subtle changes or differences, but they're statistically significant, in part because we have so much power. You can ask for each of these waves here at age 35, 60, or 80, what are the top proteins that change? And then you can look in which categories are these proteins. And again, it's not listed here, but extracellular matrix is a key feature of, for example, this latest age of change. But even here, early on, you have matrix changes. And also important to note, these are not simply sex hormones, but you have, in men, you have an equal number of changes here, suggesting really that the body shows dramatic changes at this age of around 35 years of age. Intriguingly, this is when evolution has sort of, given up on humans, because at that age, the offspring is supposed to be guaranteed, and there is no real evolutionary pressure anymore, or at least much less evolutionary pressure on maintaining the organism. A bit depressing, but that's what it is. So the exciting aspect of this is with all these changes happening, you can ask now, if you look at an individual, can you calculate the relative age based on the pattern of protein expression? And this is a concept that more and more people are pursuing now, and it's in a way it's trivial, because you can imagine if something changes linearly with age, and you have a person, and you measure what that specific measurement value is at in that person, you can basically tell them how old they are. And so people develop mathematical models. They're typically linear, called lasso or ENet models, that correlate whatever parameter you measure with aging. And just mention a few here: DNA methylation, facial features, telomere length, protein composition, metabolic changes. Again, if something changes with age and you have enough samples, you can calculate, you can generate a so-called clock, and then in an unknown sample, in an unknown organism, you can calculate the relative age of that organism. And again, this has been done by a lot of individuals, and really been pioneered by Steve Horvath by looking at DNA methylation changes, which are surprisingly precise as an organism gets older. And this is across any type of organisms he has looked at, and others. There is an accumulation of DNA methylation across the DNA, and the pattern of these changes allows you to calculate the relative age of an individual. And so we applied the same idea to the plasma proteome, and this was done in unpublished studies by Hamilton O and Jared Rutledge, two PhD students, with the idea that proteins, even from the brain, are collected into the blood. And so if you measure their relative levels, you can estimate the age of the organism. But more interestingly, you can estimate the age of a specific tissue. And so the way we do that is coming back to the tabula muris, but also to a tissue map. Tabula sapiens was just published, where you look at which genes are expressed uniquely in a specific cell type or tissue. So we don't require for this experiment it to be absolutely unique, but to be highly expressed, to at least fourfold higher expressed in a given tissue compared to all the other tissues. So if you do that, and then you look at, in this study here, we had almost 5,000 proteins. Which of these 5,000 proteins are most likely derived from the brain, because their genes are mostly expressed in brain cell types? we find almost 200 proteins that are brain specific, liver specific, and so forth. And so based on this information, and because many of these proteins change with age, we can now build clocks for each tissue. And again, the way you do that is you have the actual age of an individual, and then use these specific proteins from that, these plasma proteins. So these are, again, proteins derived from the tissue, but present in plasma. You build a clock, and the prediction is not perfect, and you would not expect that. But what people think is that the deviation, the delta H or the age gap from this perfect prediction of your chronological age contains biological information. So this person would be relatively younger, this person would be relatively older than their actual chronological age. And so again, based on this information, we build clocks for each tissue. And we had a healthy aging cohort for training. We trained an organismal clock, so we use any type of protein in plasma, or then these tissue-specific proteins in plasma. And then we validated in 2 different cohorts, again, the longevity cohort from near Barcelona, as well as our Alzheimer's research cohort at Stanford. We built these clocks. They have the organismal clock has the best overall correlation between actual age and predicted age. But again, you really want to extract the difference, the delta, and ask, is this related to biology? And so I show you here 2 examples. First, the heart. So again, the heart model was trained with heart-specific proteins. It was actually only about 10 proteins. And then we calculate in these individuals in an independent cohort, what is their relative heart age? We have people who are their heart predicted to be younger biologically, or it's biologically older. These are the different proteins. Intriguingly, we find 2 of the currently clinically validated biomarkers of heart disease. So if you go to the doctor and they measure, they want to check whether your heart is healthy, they usually measure troponin and natriuretic peptide B, and we picked out both of these. Our algorithm used these, but it also selected a few new ones that had not previously linked to heart health. And so when we then looked into these 2 independent cohorts, we find that in the Alzheimer's research cohort, we could pick out those people who had received a pacemaker. They had significantly older hearts. So if we looked who had a pacemaker before, they most of them had predicted older hearts, and the same in the longevity study. Here we looked for heart attack or bypass surgery. Again, those with heart disease tended to have older hearts. If we look prospectively in the longevity cohort, so here we had blood collection at time 0, and then there's follow-up of 15 years. Those who have young hearts, the bottom 25% heart agers, show almost no incidence of heart failure, whereas there's a 15-fold increase risk to have heart problems or heart failure in those with the old hearts, the top 25% with predicted older biological hearts. And then lastly, the same in the brain. So we predict here the brain age based on brain-specific proteins in plasma. We have people predicted to have younger brains and older brains. So these are the bottom and top quartiles. When we then look in the Alzheimer's disease cohort, who has Alzheimer's disease? It's really 18 out of the 22 in this cohort have older brains, and only 4 have predicted younger brains. And again, prospectively, here we have only 5-year follow-up, but the plasma was collected at time 0. When we look 5 years later, those predicted to have a young brain show much less incidence of cognitive decline. Here we look at the CR sum of boxes change. So an increase in this score means your cognition decreases. There's a 7-fold increased risk to decline cognitively in these individuals just over 5 years if they have an older, predicted to have an older brain. So we're very excited about these findings. And again, if you think about the bigger picture, what we're doing here is we basically look at the composition of the blood to learn about the physiological state, the health state of a tissue, of a cell, of an organism. And we can do this non-invasively. So this could potentially help you to monitor health and predict who is at risk to develop a specific type of disease. And when you do this at the organism level, for example, if you look in the Alzheimer's patients, they clearly have older brains, as I showed you, but they also tend to have older hearts, older muscle, older kidney, compared to people without Alzheimer's disease. And that in itself also contains a lot of interesting biological information, because you can now ask, what are the common proteins that are in these model? Are there biological pathways that are linked to that? Is aging potentially even contagious or spreading here across the organism? And with that, I'd like to conclude and thank all the collaborators at Stanford and other places, in particular Andreas Keller, together with Fabian Kern, for all the statistical bioinformatics analysis and brainstorming about our large data set near Barcelona, for our close collaboration with his amazing cohort, aging cohort, lots of lab members, current and former, and then funding by these different agencies. Thank you.
Thank you so much for that. That was a really interesting talk. So now if people have questions, feel free to put them in the ask a question box and I'll try and get through several of them. So one thing, I'll start with one that like made me think, well, I had a specific question regarding the CSF project and the FGF17 that you talked about, but more broadly, do you think that like this, did you identify the source of the FGF17? Was this coming from the periphery? And if so, was this in a tissue that was differentially regulated and seemed to, quote, age faster?
Yeah, that's a great question. So the FGF17 seems to actually come from a specific population of neurons that we discovered both in mice and humans. And with in situ hybridization, we could show that the relative levels of FGF17 change, they go down with aging. And so there seemed to be a connection between the production, the local production of FGF17 in these neurons, possibly making it into the spinal fluid, and then diffusing to oligodendrocytes, potentially directly through the interstitial fluid. But we were able to measure FGF17 in humans. In mice, we did not have a sensitive enough assay. It's possible that you can reconstitute that by simply infusing it into the CSF, the spinal fluid, as we showed experimentally. Whether it could come from the periphery, we don't know, but it's possible.
I see. So let me grab a question here from the chat. So Luis Leon asks, did you manage to identify markers in tissues that are more resilient to the exposure of an aged environment during the parabiosis paradigm? I think you might have touched on that.
Whether they are more susceptible or?
Oh, so if you identify tissues that were more resilient to the aging.
Oh, with the, yeah, I guess so. I mean, the resilience concept is very interesting. And I think it could equally well be that cells or tissues that do not age as much have more resilience rather than there's less factors driving or driving the aging process. And this is certainly in the context of this new initiative. We want to pay much more focus on this. You can imagine that humans, for example, are at risk of neurodegeneration, different neurodegenerative diseases, that if different cell types in the brain or different brain regions have different rates of aging, you would be more resilient to one or the other disease.
Yeah, that makes sense. One other thing I noticed from the data you showed, you had mentioned certain genes were impacted more, such as extracellular matrix, and different cell types, such as stromal cells and endothelial cells. Um, do you think that itself is just a factor? Is there a breakdown in the barrier integrity across the whole body, and maybe some organs like the brain are more susceptible in different cases?
Yeah, that's entirely possible. It's the there's a whole field that studies the matrix matrisome, I think they call it.
Mhm.
And I think it's incredibly interesting, but also very complicated to study. The brain is full with matrix, and it really changes dramatically. I mean, even if you experimentally, for example, I worked with mice, but if you isolated a young brain or an old brain, you could just feel that the texture is very different. An old brain is really stiff and hard, and the young brain is very soft. Um, and that is the matrix. And what this does, it has mechanical consequences on how cells respond, but it probably completely changes the way cells respond to their extracellular environment, having access to different factors. Diffusion rates will be changed. Really, everything I think changes.
I see. Let me see if I can grab one more question. So that's interesting. You mentioned the spread of aging across the tissues, and I forget what the word you said, but this person's wondering, do you think a pathogen may explain some of the aging processes? For example, microbiome analyses of the blood have been shown to help cancer diagnoses.
Yeah, I think the microbiome has huge effects, and microorganisms in general. Um, if you look, for example, at the specificity of T cells in our body, we all get exposed, for example, to Epstein-Barr virus, almost everybody, or to cytomegalovirus. And as you get older, more and more T cells are basically busy keeping these viruses in check. And as a response, you get sort of inflammation. Herpes viruses are another good example. Um, Many people suffer from sort of outbreaks when they get stressed. They suffer from outbreaks of herpes labialis, and then the immune system gets reactivated. You get an inflammatory response. The virus gets put down in check. Um, but those waves of inflammation are part of how we age, and they probably drive part of the inflammatory response that we see with aging, and potentially even drive the aging process.
I see. I guess somewhat related to that, do you think that this aging signature that you've identified and used to like predict things, could it also just be a disease signature? So say with someone that had a heart attack, would they look like they have an older heart?
Yes, absolutely. And that gets to the question of, what is age and what is disease? Because they're so intricately connected. It's often in the Alzheimer's field, there's often a debate, is this aging? If everybody gets the disease when they get old enough, or almost everybody, um, probably 95% of people, if they would live old enough, they would get Alzheimer's disease or some form of um, cognitive decline that fulfills the criteria of Alzheimer's disease, then, what is aging and what is disease? And that's much cleaner to separate in young people, where you seem to have a very specific disease process. But then even if people look at the transcriptional signature of these disease processes, there's often overlap with aging. Yeah.
Yeah, it's tricky to find, but
Mhm.
super interesting work. And thank you so much for sharing. I'm going to put in the chat a link for a trainees meet the speaker session if people would like to join by Zoom. That will just be for until 9:30. So please, thank you. Thanks, Dr. Wyss-Coray. And this again is the last talk of our season. We'll return in September, so stay tuned and check out our website to get on the mailing list. So thank you everyone for the great season, and thank you for this great finale. All right, I'll
Thank
add
you.
this. Great. I will, if you want to jump on the Zoom, I'll be right there too.
Okay, do you have the Zoom link? Okay, I copied.
I
Yeah,
just
okay.
stuck it in the chat. I'll
Okay, sounds good.
see.
All right,
Great.
see you there.
Thanks
Bye.
everyone.